So the purpose of this course really is to provide an introduction to a brand new field of bioinformatics. It's very much at the interface between computer science and the life sciences. What I'm going to do in today's lecture is give you some background to the biological data referred to as the field of genomics, which has produced enormous data sets that are really the foundation for the value of bioinformatics, essentially the ability to get answers to biological questions computationally. So today's lecture will be a bit different from most of the course in that we're really going to be doing kind of an intro overview of some of the underlying data and problems more on the biological side. After today, we're pretty much going to switch over to more math and algorithms, so it'll change quite a bit after today. So one thing that has changed a little bit over the years is the organization of the bioinformatics courses, and I just want to mention this in case it's relevant for anybody here. So about a year ago, we split what had been up to that time a single track for Intro to Bioinformatics, which is this course, into two tracks. One track, this course, that is intended for students who have some preparation in statistics and probability, and as a typical prerequisite, Stats 100A. Familiarity with probability, probability density functions, etc. And second, both the ability to program proficiently and familiarity with algorithms and data structures. So every other week, we're going to be having programming projects in place of our regular homework assignments. And then in between those, we will have regular homework assignments that are more solving problems on paper. There is a second track, which is offered in the molecular, cellular and developmental biology program, MCDB 172, taught by Matteo Pellegrini, which is aimed more at biology students who don't have perhaps one of those kinds of prerequisites, and it focuses much more on the problems of genomics and bioinformatics from the point of view of tools that are useful for biologists, whereas this course is very much about learning the underlying theory that allows you to invent new kinds of bioinformatics. So we have a website which I'll mention to you in a moment that gives all the course syllabus and other information is already posted. But I'll start right away by telling you about the two textbooks that we're using for this course. I apologize for the fact that there are two textbooks. It's a little difficult in this new field to find a single textbook that covers all the different aspects of the theory that really are crucial. So this really splits into two halves. One that's more of a CS textbook about algorithms, and second, what is effectively more of a statistics textbook talking about the underlying math and probability theory that's essential for many aspects of bioinformatics. The one saving grace for the choice of two textbooks is that if you do continue on to the follow-up course next quarter, 160B, 260B, I believe it'll probably have a CS numbering in sequence as well next quarter. They use exactly the same pair of textbooks. So it's one pair of textbooks for two courses if you go that route. As I mentioned a moment ago, we're going to be having homework and projects. They will be given out in class on Thursday and up on the website, and then due in class the following Thursday, one week later. I want to introduce our TA, Mike Janis, here in the back, standing room only, manning the camera. And as you know, there's a discussion section that he's going to be running on Tuesday mornings at 9 o'clock. The grading, as you see up here, this is for the undergraduate section of the course, is sort of split between quite a bit of emphasis on the projects and on the homework together, I guess, a pretty significant portion of the grade. There's a single midterm, S, and then the final exam at the end of the quarter. Graduate students are graded separately, and also there's an extra term paper requirement for graduate students.
Question?
Please.
Are these slides going to be posted online?
Yes, absolutely. You are way ahead of me. The next slide was about what exactly we're trying to do online. So we've created a website, C260A, Bioinformatics, UCLA Review, hopefully easy to remember. In addition to the sort of obvious aspects of just posting everything, such as the lectures, any materials for the course, we're going to have some additional readings in addition to the textbooks that we'll post. What we're hoping that this website will actually enable will be some kind of discussion forum type functionality. So we're using WordPress, which allows you to have kind of a blog-like format in which every single piece of information that's there you can comment on, and then we'll be able to reply by putting another comment, or you'll be able to see other people's comments, and in effect we can hopefully use this to be able to have a very fluid dialogue on material for the course. And it is spam protected. All right. So, what are we here for? Bioinformatics, what is it? Well, simple answer is that it is, in this modern age of high throughput experimental data flooding from biology laboratories, bioinformatics is the way that you answer questions in the genomics era. And I'll try to explain exactly what I mean by that with an example. So genomes, particularly, for example, the human genome, turn out to be very complex structures. The traditional picture of a gene that biologists developed from looking at bacteria is the idea that a gene is very, very simple. It's a single block of sequence with 4 nucleotides, A, T, G, C, forming an alphabet that encodes the function of a gene, producing a protein that goes and does some biochemical work. However, as you go through the sequence of major achievements in genomics, first the sequencing of the yeast genome, about 13 million nucleotides in length, about 6,000 genes packed into that, virtually all of them in this very simple one-piece structure. But going forward to slightly more complex critters, in this case a little fruit fly, about 10-fold larger genome, about double the size in genes, you'll notice that the expansion in the size of the genome seems to be outstripping the increase in the number of genes. And as you go on, for example, to human, that really becomes a serious problem for people trying to analyze and interpret the data. So now a 3 billion nucleotide total length of the genome, and now the gene structure is broken up into this highly complex structure of individual fragments called exons, separated by long intervening sequences called introns, that are actually removed from the final product. When you take a gene and activate it, what it does is it basically copies out this whole region, then it comes back in and cleaves out the individual introns and pastes together these individual exons. And you can see here's a gene already with 28 exons that have to be pasted together. The complexity of this right off the bat is for people who sequenced these genomes to even see what did we actually end up sequencing. In other words, just because you have 3 billion A's, T's, G's, and C's doesn't mean you understand what it does. Well, the first thing that you want to know about what it does is what genes and what proteins does it make. And I think you can see that it's a lot easier to recognize a gene when it's in this kind of simple one-piece structure versus needing to figure out all the fine details of what's exon and what's intron just by looking at the letters of the sequence. So when we have such a high level of complexity, one can see that immediately among biologists by seeing how they react to it. I'll just pose 3 questions as examples of why we need bioinformatics. We don't understand how all this complexity emerged evolutionarily and what it means medically. And we can only answer those kinds of questions by doing very large-scale analysis over these entire data sets and over actually many, many different organisms. So how do we know that we're operating within the domain called complexity? Well, the answer is when everybody gets really confused and can't agree with each other. So here's an example of a paper published in Top Journal a few years ago, analyzing sequence fragments from the human genome, and they estimate that there are, based upon that data set, a total of 35,000 human genes. And then here's another paper published actually in the same issue of the same journal, where now they get the number of 120,000 genes. It turns out that the answer was much closer to the first estimate rather than the latter estimate. But we have to ask ourselves, starting from the exact same inputs data and the same access to computers and analysis methods, why is it so hard to count? I mean, this is just a counting problem, right? The obvious answer is that complexity that I mentioned a moment ago, that you've got all these things in tiny little fragments, and figuring out what's what isn't trivial. And this is one reason why the real problem in genomics is actually the information problem of being able to figure out what the data mean. So another example of the need for bioinformatics arises from the sequencing of the human genome. And as I mentioned, a relatively low number of genes was finally determined from this. And this in itself raises another kind of puzzle, which is that, here we have a tiny little nematode worm. It's microscopic in size. It has 19,000 genes, really not that much different from the little fruit fly that I mentioned a moment ago. And we tend to think of ourselves as being, dramatically more sophisticated and complex than these little guys. Yet our genome, the latest kind of consensus estimates are actually even a smaller number of genes than this, maybe in the 25,000 to 30,000 range, depending upon how you count. So, what is it that actually is giving rise to what we perceive as a much higher level of complexity, for example, in our mental functions, than would be implied by the relatively modest difference in the number of genes, maybe on the order of 40 or 50%. Just to give point to what I mean by less complex, this little nematode worm has so few neurons that the biologists actually have gone to the trouble of mapping and naming each one of them so that they can then go do developmental studies in which you knock out one cell and see what happens. Okay, so the challenge that we face is that there's truly an enormous amount of data being produced through some technologies that I'm going to tell you a little bit about today. And no human being is ever going to go through all this data. So we can't do science the way we've traditionally done science. In other words, a scientist staring at the data. Actually, it's going to have to be done computationally. So what are the technologies that have given rise to this new science? This is generally referred to as the field of genomics. It's intimately related to bioinformatics, and I'm going to try to explain how these things are connected and how they differ from sort of the past 50 years of biology research, which can be generally referred to as molecular biology. So I'm going to try to define each of these terms. In general, genomics being the genome scale experimental analysis, depending upon several kinds of engineering factors, and then bioinformatics as the method of interpretation of these kinds of data. So we can trivially define genomics as sort of molecular biology to the nth power, where n is actually just the total number of genes in a genome. It's what you can do if you have a technology that is capable of measuring or characterizing every single gene in the entire genome simultaneously, ideally in one experiment. The first kinds of waves of genomics, like sequencing, obviously weren't actually able to handle all of that in a single experiment that you just do in one day. The sequencing of the human genome initially did not take a day. It took actually years. Now it is possible to sequence an entire human genome in a day if you really want to. And there are now wave after wave of additional technologies that I'll mention to you, such as DNA microarrays, etc., where you actually have the ability to measure, for example, the level of activity for each individual gene in the entire genome of that organism in every single sample for as many samples as you want to analyze on one day. So these kinds of waves of technology have really created an avalanche, orders of magnitude more data available to biologists than they ever dreamt of before. And that process is going to just continue wave after wave after wave. This is not something that's done, it's something that's just started. So what's it like to go from approximately nothing to approximately everything over a couple of years? So when I started out in biology and was going to graduate school, which really wasn't that long ago, we were all taught the central dogma of molecular biology, which is that one gene equals one protein, which then goes and does one specific biochemical reaction. And one can get sort of cynical about how molecular biology research typically is done, which is the sort of central dogma of molecular biology research is that one gene equals one laboratory. In other words, one research group. All they do is work on one gene for the rest of their lives. And then the next level is that you go and work in a lab and they tell you, okay, for your PhD, what you have to do is go clone a gene and then sequence it and write a couple of papers about it. So we only have the one gene, one PhD pattern, which was very common when I was in grad school. And relatively recently, a graduate student here at UCLA sort of took this to the next level, which is, she sequenced one genome, so okay, so she gets a PhD. I think you can see sort of the direction that this is going. For something like 50 years, molecular biology and research in biology in general meant cloning, discovering new genes. What does it mean for a science when now essentially all of the genes are just sitting in a computer, in a database, anybody can download them from the internet? That's what happened over the last few years. Say 10 years ago, we had about 20% of the genes studied enough such that someone had given them a name, which doesn't really mean that much, but at least somebody knew they existed. And now we basically have everything. So you go from 20%, which is sort of like nothing, to essentially 100%, and now we have hundreds and hundreds of complete genome sequences. The way that was done was essentially going from a tradition in biology, which was a lot like cooking. In other words, you had some ingredients, you had a cook, you stood basically at a kitchen counter, and you mixed your ingredients, albeit in microliter quantities. But over a period of a few weeks, you could whip up enough recipes that you could sequence, say, several thousand bases. The main difference from cooking was you couldn't eat what you made. Engineering has really replaced that, so that you replace exactly how you read the data instead of using photographs of radioactively labeled bands on a gel. Instead, now you're using fluorescence dyes that are read in real time with a laser and a high sensitivity detector. You can generate orders of magnitude more data per read per minute. And what this really is just plain old engineering and technology. There are many different levels to that. Eliminating every step that actually depends upon a human being. Multiplexing every single reaction so that instead of doing one reaction at a time, you're doing a minimum of 96 or 384 reactions in one little plate. You're miniaturizing so that instead of working with that much material at a time, you're working with truly minute quantities of material in each one of those little wells. You're raising by orders of magnitude the sensitivity and the speed with which you can read out the information, and you're effectively adopting kind of an engineering point of view instead of a cooking point of view about every single aspect of the reproducibility of the system. And through this sort of technology, people have been able to completely revolutionize and raise by orders of magnitude the data production. So even at the beginning of this, it obviously had computational problems. Here's an example of the physical format of a sequencing gel. What it is a very thin film in which you load samples at the top. Those samples are driven by an electric field down through the film, and they separate on the basis of size. And you use 4 different fluorescent dyes that have different colors of fluorescence, such that each product in that mixture gets labeled by the appropriate color, indicating which nucleotide is the last nucleotide in that product. And so actually by just reading up one of these lines, red, green, green, red, blue, etc., you can read off the nucleotide sequence. You can see that even with this fairly simple problem, you have all sorts of issues of being able to separate the different lanes from each other automatically. You can see that it doesn't quite run straight here, it starts to bend a little bit, so various kinds of mapping would be required to be able to completely automate this as an image processing analysis. So you have to track the lanes, you have to extract the intensities, you have to interpret those intensities, and oftentimes you'll see that there are sub-peaks where things start to mix and you're not getting clean separation of the signal, but this is about as good a quality gel image as you'll ever see. So actually the top peaks are enormously above the noise. In real sequencing, it's not like this. Oftentimes things get quite mixed up and you have to really be able to estimate the probabilities for what's the true nucleotide sequence and incorporate that into high throughput analysis that uses some multiple runs to establish the correct sequence. And what this has led to is, for example, at the Whitehead Institute at MIT, sort of the new face of biology is that lots of robots and very, very few people. It's not cooking anymore. It's more like a factory. So in this way, people have been able to scale up from the first genome sequencing, which was completed at the Institute for Genome Research by Craig Venter. I think the day before he received a grant rejection from NIH saying that it couldn't be done, through a fairly rapid progression of orders of magnitude increase in sequencing speed to the point where now, today, we can effectively sequence the size of the human genome in a day, and it's actually not that expensive. So what this has led to, really, is a dramatic change in the nature of what actually counts as science in this process. In the past, experiments were designed in a very, very targeted way, and if you couldn't immediately understand what your experimental result meant, the moment that you picked up the, you looked through the microscope or looked at the gel to see what the result was, it meant you didn't design the experiment right. Just didn't think through what you were doing. By contrast, in genomics, you know, what is the hypothesis that is being tested by sequencing the human genome? It's just sequencing, right? It's not a question being asked in a very targeted way. Instead, what it is a bet that if you collect all of this data, you're going to learn an enormous amount about human biology. It's true, but what it means is that it is no longer the case that the science is done in the design of the experiment. Now the science is done basically in answering the information problems, analyzing what is the actual information content in the massive amount of data that you've collected. So it really becomes a computer science problem, both of finding patterns within a data set and also a problem of evidence for measuring the strength of evidence that those patterns are real and not just noise due to the fact that you searched really hard over a huge data set. Of course, you can find patterns if you want to, even if the data was completely random. Okay, so trivial example of that is the sequencing of the human genome. It was a hard problem, 3 billion nucleotides divided up into all sorts of little pieces. The main thing I want you to take away from the technology that was used to do the sequencing is that it was actually relatively inexpensive just to grab a random piece of DNA, put it into the machine, and get sequence out. The thing that's really difficult is that the machines could only sequence tiny little bits at a time. You could get out like maybe 500 nucleotides at a stretch, one little fragment. How do you sequence 3 billion nucleotides when all you can do is sequence it 500 bases at a time? Well, I think you could see that if the text were clear enough, you know, if we were basically taking a Shakespeare play and chopping it up into tiny little pieces and then reading each one of those pieces, and we could read, you know, each different section of Shakespeare multiple times, we might be able to kind of put it together as a jigsaw puzzle, you know, in one dimension and see, oh, okay, this fragment of text seems to overlap that fragment of text, and so we can basically piece together what the full text is. The problem is that the human genome actually has a lot of redundancy in it. There are large regions of the genome where one spot in one chromosome actually looks letter for letter identical to a long, long stretch that could be thousands or tens of thousands long in another stretch. So that really defeats at least a completion of the genome sequence in that kind of way. So what this really shows is that the experimental part of sequencing the human genome, in some sense, was the easy part. It was what some people solved first. It was actually the information problem of being able to put Humpty Dumpty back together again, which is referred to as the assembly problem, that really people struggled with. So that's shown kind of pictorially here. You've got this massive, huge object that you're really trying to sequence. You can't sequence it in this form. You can only sequence it by busting it up into pieces. And then actually, even to sequence those pieces, they're too long. So you have to bust it up into even tinier pieces. And then you get these pieces sequence read. And then you look, oh, this TGAT, CAT seems to match over to this end of that fragment. So you put those two together, and that's assembly. The company Celera actually bet that they could assemble the entire human genome sequence this way, and they ran into trouble when they actually tried to complete the full assembly. They actually had to make use of public data. They were competing with the public sequencing project that was using a different approach that had more mapping data. And when you put all these things back together again, for example, take the actual sequence as it was assembled by a particular assembly program, and now you compare it against some completely independent experimental data that basically is a map that biologists have built up through genetic studies over the years. You can see, oh, it's a pretty good fit, looks linear, But then you can see, for example, here's an error where things seem to be off. Here's an inversion where a whole segment is actually backwards. You can see it's flipped around here relative to what it should go track as a linear increase. So the only point that I want you to take from this is that actually it was the information problem of assembly that turned out to be the hard part. Of course, it just got worse going forward from there. Once you get all this sequence, 3 billion letters, what are you going to do with it? What does it mean? Well, the first thing that biologists want to know from a genome sequence are the so-called genes. These are the active, functional portions of the genome. And your initial ability to find genes within the sequence depends upon how complex the gene structure is. In simple organisms like bacteria or even yeast, as I said, the gene structure is simply a single block, and you can use some statistics that we'll study in this course to predict with high accuracy, okay, this looks like a gene. Unfortunately, as you saw in the human genome, that's not the case. Genes are broken up into a very complex structure, and the initial efforts to use those statistical prediction methods to predict the genes straight from the 3 billion letters of the nucleotide sequence actually did a pretty bad job. And something like half of the reported genes were either completely wrong or significantly wrong in important details about what the actual boundaries of exons were, which really changes the result. So the bottom line, even the next stage of so-called annotating what are the genes, again, turned out that the information problem was the harder problem, and the one that really mattered for being able to make sense of the meaning of the data. So the genome annotation had to be done computationally simply because of the scale, but yet at the time that all of this was initially done, the computational methods, statistical methods weren't up to the challenge. So these are all examples of bioinformatics being the crucial ingredient for actually extracting some meaning from the genomics data. And for myself, I define it as a kind of an abstract definition of inherent structure present within biological information. That is to say, when we look at something like the human genome sequence, actually it is obvious that there are some patterns in that data. I mean, even just looking at the simplest kind of Markov chain or triplet frequencies or what have you, you can see big patterns of variation within the data that are clearly non-random. So those kinds of things actually allow you to identify genes, to identify exons pretty immediately just from staring at the data. Of course, there are many more layers to this problem that are non-obvious, and they extend all the way out to the fact that that genome encodes how to build the whole human body and the functioning of the human mind, which we clearly don't have any kind of handle on yet. But fundamentally, what I'm proposing to you is the idea that bioinformatics is kind of the full stack of those layers of structure in the biological information. And ideally, we would let the data speak for themselves by basically exposing non-random patterns within the data, which of course requires us to have measures of evidence for any kind of patterns that we try to dig up from this data. So some examples of these kinds of things are to be able to identify which genes are important for controlling the cell cycle, which is basically the cycle of stages of growth in a cell. There are different stages, such as the stage where it copies all of its DNA. So for example, there have to be tons and tons of genes that are involved in DNA replication. They all have to become active right at that moment in the cell cycle. And people have used bioinformatics analysis and genomics data, where they essentially synchronize a whole sample of yeast cells so that they will all cycle in synchrony with each other. You take a sample every 5 minutes. You take that sample, you extract the RNA that shows which genes are active. You analyze that on a microarray that essentially has a separate spot for every single gene in the entire genome, and the intensity of fluorescence at that spot measures for you how active that individual gene is. And you measure that for every single gene in every single sample at every single time point. And what you see are curves going up and down, and then you extract those curves, cluster those based upon their expression patterns, and then you can look, because you have the genome sequence for every single one of those genes, you can actually look at its sequence. If you see a set of 100 genes that are all being expressed in synchrony with each other, they turn on at the same time, they turn off at the same time, you say, well, what's controlling these genes? We know that the DNA sequence immediately upstream from the gene typically is the most important region for controlling when it turns on and when it turns off. So we can extract those regions of the genome sequence and ask, can we find individual short sequence motifs that are found much more frequently in this set of genes that are all expressed in synchrony compared with all the other genes in the genome? Please stop me if you have any questions. So this sort of problem is often a modeling problem where we have to decide what kinds of parameters we're going to extract from the underlying data to relate this back to other kinds of computer science problems that we already have solutions for, the computational complexity of the problem such that we can solve it in a really effective way, both in terms of time complexity and memory complexity. And one basic thing that I want you to draw from the overview of genomics is that it really represents a major change in how biologists try to answer questions. So in the past, we were operating very much kind of in the mode of exploring a very big, very dark cave with, one of those tiny little pen lights, flashlights that only can illuminate a tiny little square right in front of you. To answer any kind of question, you had to take into account the fact that you just couldn't see what was going on with 99.99% of the genes in that genome. You didn't have a method that could actually measure them all at one time. So you had to concentrate your effort on just a few genes at a time. So what you would do is design an experiment that is clever in that it can actually answer a question by ignoring most of what's going on. In genomics, actually cleverness is the exact opposite. It's what becomes possible only when you have a complete data set. So complete might mean we measured the level of expression or activity of every single gene in the genome. And that's a hard thing to get your mind around, at least for biologists. So I'm going to give you a simple example that hopefully will illustrate for you the power of this way of thinking. And then you'll see some examples during this course of how we use this kind of idea. All right. So first, a definition. Many genes in each species genome. So mouse genome, the human genome. We know that we're highly related to each other, and when we sequence the DNA, we can see that. So what should we call a gene that is found in both human and mouse that does exactly the same thing, and which by sequence we can see are highly related to each other? Well, the biological term is an ortholog, and we would define that as two genes that are related to a common ancestor that they descended from solely through a process of speciation. By speciation, I simply mean that two daughter species were derived from an ancestor species. So an ortholog we define as two genes that are related to each other solely through that speciation process. Nothing else has happened on the tree of relatedness other than speciation events. By contrast, we also see another kind of relatedness in genes between different genomes that I'll refer to as paralogs. And the idea here is that biology is parsimonious. It creates new functions not by completely new invention, but typically by just taking some existing thing, copying it so that you now have two copies, taking that extra copy and rejiggering it a little bit. And we see this all the time in genomes, and this is what we're calling paralogs. The extra step that we've added is that these are two genes that are related by at least one gene duplication event, and these gene duplication events are always tied with a very increased speed of divergence or mutation, where the genes change much faster from each other because of the fact that you have two of them. There's not as much pressure at maintaining function of at least one of those genes because you've got another copy. So paralogs related by at least one gene duplication event. Okay, now we sequence a brand new genome. We find sequences that are similar, say, to the human genes. And for any given pair that we find that are similar, is it an ortholog or is it a paralog? That's a very difficult question experimentally. Biologists would actually argue in the past for a long time. And that argument actually mattered. For example, you can use a mouse or even maybe one of those tiny little nematode worms as a model system for studying a human disease if you know what human gene is involved in that disease and you know the ortholog in mouse. Oh, you make a mutation in that gene in mouse, and you now have a mouse that you can study to figure out how to cure that disease. So knowing what's an ortholog versus a paralog is crucial information. It's actually also the cause of unwanted side effects or lack of efficacy in drugs developed for human diseases. Think about it. Say you have two highly related genes, paralogs. You make a drug that targets one of them without realizing that the other one is there. You're going to screw up the other one as well. That could cause side effects. Alternatively, you develop a drug for one of them. You don't realize the other one is there. This one blocks that target gene successfully. Oh, but the other gene is a little bit different. It's still working. So your drug has no effect because the other gene is still carrying on the related function. So these kinds of questions, orthologs versus paralogs, have plagued medical research for decades. So let's look at a simple example of how we could solve this problem computationally in the last few minutes here. So what I'm going to draw here is a tree structure showing how orthologs and paralogs would be defined for an example evolutionary tree. So let's say we have an ancestral gene shown here by this triangle at the top, and now various events happen. So for example, here we have a speciation event shown by these square brackets. So that means now this species divides into two separate species, and then let's say that happens again. So down below here we have two different species. They each have a copy of the gene. That gene is orthologous between those two species. It's just the same gene, just in two different species, connected by simply a speciation event. Is that clear? Okay, let's say we add one more complex, one more complication to this process. I show a gene duplication event with this angle bracket shown right here. That means what used to be a single gene in this genome now has two copies. And of course, when that genome splits into two different daughter species, both of those copies are going to get carried along in both of those species. And so now what we have are two sets of genes in each species. So we have a pair that are orthologous, the triangles, another pair that are orthologous, the diamond, and that's in one species versus the other species, but they are paralogous to each other, and this triangle gene, for example, is paralogous to this diamond gene. Why? Because they're connected through this gene duplication event. So, as I said, this was very, very difficult to sort out experimentally. People would literally spend years figuring out whether one particular gene really was an ortholog. What if we had a computational method that could, in one run of a computer, analyze all genes in many genomes and figure out which ones are orthologs of each other and which ones are actually paralogs? So there actually is a quite simple way to do that. As I told you a moment ago, every gene duplication event gives rise to a period of rapid divergence, much more rapid mutation rate. So the simple message of that is, if you compare orthologs versus paralogs in the same pair of genomes, which of course are separated in time by equal amount of time since their common ancestor, split from that common ancestor, the orthologs will be more similar to each other than a paralog will be to another paralog. So when we search by some kind of sequence similarity method, what we would expect is that if we have gene A in genome 1, we search all of genome 2, we would expect that the sequence that matches it with the highest level of similarity should be its ortholog. There might be other genes that show some similarity, but we would predict that they would be paralogs. And even better, we would expect that if we took that candidate ortholog and we used its sequence to search all of genome 1, that best match should come back reciprocally to the gene A that was matching, that found it as the best match. In other words, we should have a best match in both directions bidirectionally, as shown here. So we take gene A, we search all of this second genome. It finds something as its best match. We take that gene, we search against all of the genes in this genome, which could have matched any of these as the best match. But if they match reciprocally to each other, that predicts that they are orthologs. So we can basically take these kinds of trees that we would get. So these are orthologs. Here's a gene duplication event that creates a paralog. And now we draw the best match relations as either bidirectional, if it's a reciprocal relationship, or as a directed edge, indicating that this gene found that gene as its best match, but this gene did not find that gene as its best match in that genome. So from this tree, ortholog versus paralog, what we would actually expect to get from our sequence similarity graph analysis is a clique of reciprocal best matches that basically marks out that these three genes are orthologs. And then we would see similarity edges here that are directed and not bidirectional, indicating that this gene is actually paralogous to these other genes. And we can perform this analysis on slightly more complex cases. The bottom line is you can run this on the complete set of all the genomes that we have available in one computer run and actually assign orthologs over all of the genes in the genome. We'll have some cases where it's unclear, but this is a radical transformation in what biologists were able to do before for discovering what really are orthologs and what are paralogs, which matters for things as diverse as drug discovery and model organisms for disease study. So this is work that has actually been quite important. I think I will end there for today. And at the back here, you have the syllabus and the first problem set. They will also be posted on the web, and I will post lecture notes, and we will have links for the video at the talk as well. Any questions? Please come talk to me.